Dear poster visitor, thank you very much for stopping by. My name is Elena, and I will guide you through my poster, Cell Monitoring Using Multi-Frequency Impedance Recordings for Label-Free and Time-Resolved Cell Response Analysis. Reading the impedance of planar gold film electrodes that are used as growth substrate for adherent cells reveals changes in cell morphology. Thus, real-time impedance data provide insights in various cell phenotypes, such as cytotoxicity, even over prolonged periods of time. A crucial advantage over standard assays is the non-invasive continuous cell monitoring. Advanced information content is obtained by using multi-frequency impedance readouts, as they allow zooming in on changes in membrane topography, cell-to-cell or cell-to-matrix junctions, deconvoluting the complex whole cell response, for instance, to G-protein coupled receptor activation. The multi-frequency character of the data allows selecting the most sensitive frequency for a particular application or cell type after data acquisition is complete. Figure 1 depicts the cardiac site 96 system, initially made as a hybrid system recording both contractility and electrophysiology of intact cardiomyocyte networks. Here, next to electrophysiology, contractility, and viability recordings, we introduce multiparametric cell monitoring with the new software tool, Spectra Control, running on cardiac site 96 and not only for cardiomyocytes. In addition to high-quality consumables and controlled temperature and environment, the system comes with an automated liquid handling system for cell seeding, compound applications, and medium exchange. Multiple functional assays can be combined in a single plate. Transparent NSP96 sensor plates shown in Figure 1 are compatible with standard plate readers and automated instrumentation, thereby giving you freedom to combine your assays in one plate. On Figure 2, principles of impedance-based assay technology are highlighted. The frequency-dependent behavior of a cell-free and cell-covered electrode, presented in a so-called Bode diagram, A, reflects the impedance increase due to the cell layer between approximately 1 to 100 kHz. For the experimental setup presented here, the signal is dominated by the paracellular cell layer resistance between about 1 to 10 kHz, range indicated by light blue flame, and is increasingly influenced by capacitive currents across the cell membranes between 10 to 100 kHz, range indicated by darker blue flame. Panel B shows schematic side view of a cell-covered electrode. Impedance describes the electrical resistance in response to an AC signal. It takes into account that capacitors become more conductive with increasing AC frequency. Thus, impedance spectroscopy that uses AC signals with individually modulated frequencies within a spectrum enables a more diversified study of the passive electric properties of the cell layer. Examples of cell proliferation and cell toxicity experiments are depicted in Figure 3. Panel A depicts base impedance measurements at a single fixed frequency, such as 10 kHz, which reveals cell adherence, proliferation, or cell death. Hereby identified cell-specific profiles of different growth and cell behavior pattern are example relevant in cancer research. Panel B depicts exemplary cell death experiment, effects of Escin on the impedance signal of CHO cells in various concentrations, as well as the effects of Triton-X 100, 2%, is shown. Data are normalized to the respective compound reference without cells and the compound addition time point. Frequency spectra reveal a so-called sweet spot. And in Figure 4, we pose a question: which frequency can spot the highest impedance change? Average relative changes of the impedance signal with associated standard deviation due to cell proliferation of CHO, A, HEK, B, and HeLa, C cells with initially seeded cell densities of 8, 16, and 32 K per well over a time period of 72 hours are shown. Data is normalized to medium reference without cells and to the time point of cell addition to wells. Frequency spectra at time points 12, 24, 36, 48, 60, and 72 hours for CHO cells with initial cell densities of 2K for D, 4K for E, and 16K per well for F. Data is normalized to medium reference without cells and cell addition time point. So-called sweet spots are marked with X and represent the maximum of amplitude change. When focusing on barrier function research, GPCR agonists relate to changes in barrier properties as shown in Figure 5. Frequency-dependent impedance recordings are the basis for many applications of impedance technology in cell analysis. Panel A shows simulated impedance spectra illustrating cell type specific dielectric properties of listed cell lines for a 0.6 mm recording electrode. Additionally, we were able to monitor GPCR activation in various cell types by applying specific agonists in dose-response studies, as shown in panels B and C. Here, we were able to monitor GPCR-mediated signal transduction by applying the endogenous agonist, dopamine, in dose-response studies, or receptor-independent agents that are used for pathway deconvolution, forskolin. In conclusion, electric cell substrate impedance sensing with frequencies ranging from 0.1 kHz to 100 kHz are successfully integrated in Nanion cell monitoring technology, CardioExcite-96 or FlexSite-96. Continuous and multi-parametric cell monitoring has an advantage over time and cost-intensive individual endpoint analysis. Nanion's experimental setup reveals the kinetics of cell behavior and allows an in-depth mechanistic understanding in real time. Multiple research areas, such as cell signaling, cell proliferation, virology, barrier function, cytotoxicity, wound healing, or immuno-oncology, benefit from impedance recordings at a frequency spectrum. For more information, please join my chat during poster presentation or contact me directly at elena@nanion.de. You may find more information on cell monitoring as well as on automated patch clamp or membrane transporter research by visiting www.nanion.de. Thank you very much for listening and enjoy SOT 2021.